Hello and welcome to Chapter 8, Part 1C, White Blood Cells and Red Blood Cells. Now, last video, we sort of talked about plasma, we talked about tissue fluids and lymph, and all to do with this 55% component of blood. Today, we're going to talk about white blood cells and red blood cells. Under white blood cells, there are two major types, phagocytes and lymphocytes, and red blood cells is just red blood cells. So before we start, white blood cells and red blood cells are both made in the bone marrow, okay? All of white blood cells and all the red blood cells in your body is made in the bone marrow from the same type of stem cells. So this is a stem cell, and if you remember in chapter 5, when we learned about the stem cell is an undifferentiated cell that can divide limitlessly, can divide many times, and give rise to a differentiated cell and also renew itself. You can go back to chapter 5 to revise this part. So the cell in the bone marrow, the stem cells in the bone marrow, can give rise and differentiate to all these different kinds of cells, including white blood cells, which include many types, red blood cells, and platelets. So let's delve into white blood cells first and then red blood cells, okay? And it's like sort of a category by category thing. So under white blood cells, also called leukocytes, they are also made in bone marrow, and there are two types. There's phagocytes and lymphocytes. Now it's important to note that white blood cells are part of the immune system, and their main function is to fight diseases. So this chapter would overlap a lot with chapter 11, and I have moved some things here to give you a little introduction of what's going to come, a little taste of what chapter 11 is like. Okay, so there are two white blood cells here, two types, which are the major types, and then under each of these types, there will be more types as well. So two types, phagocytes and lymphocytes. These are big groups of white blood cells, and under them you have specific other ones. For example, phagocytes, we learn about neutrophil, monocyte, we'll talk about them more later. And lymphocytes, we have B and T lymphocytes, which we'll talk about later. So under white blood cells, let's first talk about phagocytes. Phagocytes are produced throughout life, so you keep producing them at the bone marrow throughout the life. They are cells that undergo phagocytosis, which we have learned about in chapter 4, right, under phagocytosis. And there's this idea that, the cell can recognize what the bacteria is or what the pathogen is. We can recognize that the pathogen is not a cell of our own body by some sort of receptor. And then that cytoplasmic extensions form or pseudopodia form, and it fuses at the end, forming a phagocytic vacuole or just a vacuole. And this gets digested when lysosomes fuse with them and digest the bacteria. And as a result, the bacteria is killed. Now, bacteria is a very specific pathogen, actually. So pathogen is just anything that is non-self in general. Can be fungi, can be virus, can be bacteria. So we use the word pathogen here. Okay, so phagocytes, phagocytosis, please go and revise. Now, the function of phagocytes in particular, different from lymphocytes, is that they patrol blood tissue and organs, and they remove dead cells and pathogens. Think of them like the police, which are having roadblocks or actually are driving the police cars around trying to see if there are anyone who breaks the law. Now, phagocytes are the same, right? They patrol the blood tissues and organs, and they purposely detect what is non-self, okay, what is not ourselves, and they remove it by phagocytosis. I am so glad the police do not eat us or engulf us in any way, whereas the phagocytes actually do that to whatever that is determined as non-self. Now, we say that phagocytes are involved in non-specific defense. What does that mean? This means that it can respond to many different types of non-self antigens. Its receptor, right, can respond and detect many different kinds of non-self antigens. And it doesn't matter if the pathogen's a bacteria, a virus, or a fungi, or some dead cells, they are going to detect that and engulf it and destroy them. Yeah, so let's talk about the appearance a little bit. The appearance is a lobe nuclei. So usually nuclei or nucleus is very round. Nuclei is plural of nucleus here. And lobe means it is not a specific round shape. It has like places where it's more thin, places where they are more fat, like that. So it's a lobe nuclei. And they also have granular cytoplasm very often due to many vesicles. Well, it has a lot of vesicles because obviously it's going through phagocytosis, and phagocytosis involves formation of vacuoles and many lysosomes that contain hydrolytic enzymes. So you would expect that of phagocytes, wouldn't you? So let's talk about the first type of phagocytes here. I'm not sure why the words are not coming out. Give me a sec. Right, now, I fixed it now. So this is one type of phagocyte. So as I said, phagocytes is a big group of cells which undergo phagocytosis. Now, let's talk about neutrophils, which is one of those many phagocytes. Neutrophils have a multi-lobe nucleus, so they are not like, they are extremely strange nucleus here. You can see there's 1, 2, 3, 4 lobes. They have receptor proteins on its membrane. So don't forget, when it comes to phagocytosis, there must be receptor proteins to determine whether something is non-self or self. And if it's a pathogen, it should be identified as non-self, and phagocytosis can occur. Now, neutrophils in general is unique in the sense that when there's an infection, large numbers are released in the bone marrow. When you need it, then its large numbers are produced and they accumulate at the site of infection. They are like the emergency, like when someone calls 999, right? When there's an infection, they rush to the site of emergency and they solve the problem. But they sort of like short-lived, and they die after they digest pathogens. So really, they are a lot like suicide bombers. They engulf the pathogen and they die along with it, destroying itself and the pathogen with them. And actually, when you see pus coming out of your wound or your pimple, whatever, those are actually dead neutrophils that have died from trying to rescue you. How strange. Anyways, that is neutrophils for you, and that is a type of phagocyte. Let's talk about another type of phagocytes, or actually two types of phagocytes, monocytes and macrophages that have a relationship with each other. Now, both of them have a lobe nucleus, and it's kidney bean shaped. You cannot see here, but they are lobe nucleus. They are larger than the neutrophils by a little bit. And similar to neutrophils, they do have receptor proteins on its membranes to identify pathogens as non-self. But different from neutrophils, they are only produced when needed. But monocytes and macrophages are those who patrol around the tissues. They are those police in motorbikes or like on police cars, like driving around, making sure everything is safe. And when they are circulating in blood, that is called monocytes. But when they enter into blood, when it enters into organs and leaves the blood and enters into tissues, those are called macrophages, and that is the more mature form. So monocyte would actually mature into macrophage. Monocytes are found in the blood, macrophage are found in the organs or tissue. And contrasting from neutrophils, these are actually long-lived cells, and they are there always. Okay, they're always there in liver, lungs, spleen, kidney, lymph nodes to make sure you are safe. So that is phagocytes. So far we have learned that phagocytes have three different types, three different examples actually. There is neutrophils, monocyte, and macrophage. Let's move on to lymphocytes, which is the second major type of white blood cell. It is also produced in the bone marrow, but unlike phagocytes, they are produced before birth. So when before birth, you have already produced many lymphocytes in the mummy's womb. Now, the function is slightly different. They are involved in specific immune responses. So these are the special task force, the special team, right? And they only respond to specific non-self antigens. They won't simply react to any type of bacteria or any type of virus. Only a specific type of bacteria, specific type of virus, specific type of non-self antigens. Mature lymphocytes would circulate in the blood and lymph, okay, and accumulate at sites of infections. But again, they do not undergo phagocytosis. They are defending the body using other means that we will cover in more detail in chapter 11. Now let's talk about the appearance, because you need to be able to identify these cells under the microscope. The appearance is quite different. It's smaller than phagocytes. It looks about the same size, just a little bit larger than red blood cells, but it has a very large round nucleus here, and very little cytoplasm. See, large round nucleus, very little cytoplasm. Large round nucleus, almost nothing for the cytoplasm. Very little. In general, there are both two main types. Both are made in bone marrow, but mature in different places and have different functions, and that influences its name. So number one, you have B lymphocytes here. B or B cells, we call them. They mature in the bone marrow. So they are made in the bone marrow, mature in the bone marrow, therefore B stands for bone marrow, makes sense. And B lymphocytes are in charge of producing antibodies. Number two is T lymphocytes, or we call them T cells. They are also made in the bone marrow, but T lymphocytes mature in the thymus, giving rise to the name T cells. T stands for thymus. These lymphocytes do not produce antibodies, okay? They do not produce antibodies. They defend the body with other means that we will learn in more detail in chapter 11. But the point I'm getting here is, hey, there are many types of lymphocytes. These are two main types, but there are actually even more types. Both work together, and many others work together to defend the immune system. So white blood cells is actually a huge group of very diverse different kinds of cells, and these are just the main types. Now, as I said just now, you will need to be able to identify what these cells are and how these white blood cells look like under the microscope. So usually it's a drop of blood and you just smear it across. Sometimes they put some stains, sometimes they don't, and you get this kind of appearance. Again, it's very important to know what is what. Okay, like multi-lobe nucleus, you know that it is definitely neutrophils. Singly lobe nucleus, you know it is monocyte, and this is found in blood, so it's definitely monocyte, not macrophage. And these here are lymphocytes, which have very little cytoplasm and a very round nucleus. So yeah, identify them. If you don't know how to differentiate them, feel free to PM me for more help. Now, we also need to differentiate white blood cells from red blood cells. Obviously, it's very easy to tell because they are weird looking and they don't look like the typical biconcave shape. But how do we put that in words? So here's it in words. White blood cells are different because they contain the nucleus, right? What red blood cells do not contain nucleus. White blood cells are mostly larger than erythrocytes. Erythrocytes is another word for red blood cells, except for lymphocytes. Lymphocytes is about the same. Number three is that it is spherical or irregular in shape. Okay, not in a biconcave shape. Number four, phagocytes in particular have granular cytoplasm. Lymphocytes don't, but phagocytes, they do have this granular cytoplasm, which red blood cells do not have. Which brings us to our next major type of blood cells we're talking about today. We are done with white blood cells. Let's talk about red blood cells. Again, red blood cells, there's this different name called erythrocytes. They are short-lived. They only survive for 120 days. So blood is being produced, red blood cells are being produced every day in your body. Now, its function, of course, is to transport oxygen to body tissues. And how are they adapted to that? So remember, one of the favorite questions for CIE is that how is its structure related to its function? So here is how. Number one is that it's small and flexible. Diameter is around 6 to 8 micrometers, and you do need to memorize this. And they are able to, this enables them to squeeze through the capillaries, which are 7 micrometers. Now, because of like the sizes here, and how it's able to squeeze through, and you can see here it's so close to the outside layer here, this decreases the diffusion distance between the riblet cells and its environment. Number two, it has a biconcave dish shape. This increases surface area compared to a sphere, right? And this is for more diffusion of oxygen to the cells. Number three, there is no nucleus, no mitochondria or ER in the riblet cells, and therefore you can expect there's no transcription, that riblet cells are not dividing on their own. They have to be produced in the bone marrow from the stem cells. They don't go through any form of active transport that requires ATP. There's no ER, so they cannot synthesize stuff. But they have a lot of room for hemoglobin, which are synthesized in the stem cell. When they are being produced in the bone marrow, not in the stem cells, sorry. So if they don't have all this and they are synthesized in the bone marrow already, there is more room for hemoglobin. They won't produce any more when they're mature and circulating the blood. But before that, they would have, oh my gosh, a lot of room for hemoglobin. This maximizes the number of oxygen carried by riblet cells. The more hemoglobin you can carry, the more oxygen you can carry per riblet cell. Again, riblet cell is a cell, and hemoglobin is a molecule, and you can have a lot of these polypeptides in your riblet cell. So this hemoglobin is found in large quantities in riblet cell. Just a reminder slide for you. Now, remember that hemoglobin is made out of protein. It is a globular protein, which means it can interact with water. It is soluble in water. Okay, all the things about globular protein applies here, including the fact that hydrophilic R groups are on the outside, whereas hydrophobic R groups are on the inside. Hemoglobin is made out of 2 alpha globin chains and 2 beta globin chains, which makes it a quaternary structure. Okay, so it has 4 chains in total. Each chain has 1 heme group, which has an iron 2+ ion, and each heme group can bind to 1 molecule of oxygen each, which means 1 hemoglobin here can bind 4 molecules of oxygen to form oxyhemoglobin. And this formation, this chemical equation here, obviously happens in the lungs, where gas exchange occurs, and you breathe in fresh air of oxygen for binding to the hemoglobin to be transported to the rest of your body. So that's it. We have talked about white blood cells. We have talked about red blood cells. Just a slight recommendation for procrastination here. If you want to feel productive while procrastinating, you can go check out the anime called Cells at Work, which this character is from. It's all about how cells work in the body. It has to do with the immune system and would help you understand some things through a very fun way. Besides, it's sort of like silly, but it's cute, and it's like I learned something, but I'm chill. So yeah, definitely go and watch Cells at Work. Recommend it. I've watched 1 season so far. So yeah, hope you stay sane. Bye. See you next video.